Thank you for your time and attention today. My name is Zach Hildenbrand. I'm with C4 Laboratories, and today we're going to talk a little bit about cannabis research and where it has been and where it is going. I always find it rather imperative to talk a little bit about my journey in this field of research because it is rather unconventional, and it'll probably give you a greater context as to where we believe that this field is going. So I'm a cancer biochemist by training. I started working on hormone-dependent cancers from a structural perspective, and then I went into working on a treatment for chronic myeloid leukemia at UT Southwestern. And again, that was actually relatively unconventional research because we were actually using arsenic to cure kids with cancer. Most people think that arsenic is bad news bears, but if you use it appropriately at the molecular level, it can do quite wonder. And then, now that brought me into this field of research. And when I got into cannabis research, I kind of looked around, looked at the peer-reviewed literature. There wasn't a whole lot there. And what I would say is that it was primarily what I call bro science. Now, bro science in cannabis is, okay, we grew this plant. What do you think the THC content is? Go ahead and try it. Oh, I think it's 20%, bro. How do you know that? Oh, bros know, bro. Bros know. What should we use it for? Oh, pain management. How do you know? Well, just bros know. I know. Come on, I know. So that, unfortunately, is a terrible affliction for myself, coming from a traditional research background. We're using a lot of advanced techniques, spending a lot of money, and getting down to very, very finite resolution on complex issues. Bro science is just completely unacceptable. And so I really wanted to be a part of a movement where we could bring cannabis research out of the primordial ooze and get the attention that it deserves. I think we can all agree here that cannabis is uncharted waters. And as far as the next wonder drug, the next compound that's going to be the cure-all therapy, I think that cannabis holds a lot of those secrets, and it's about time that we spend a lot of resources, time, and energy, surround ourselves with intelligent people, and unlock those secrets. So that's what we're dedicated towards doing. I'm going to talk a little bit about here, starting at the beginning. We'll get into cannabinoid biogenesis. A lot of fun stuff here. The queen bee, if you will, is CBGA, right here. Okay, so from CBGA, the cannabis plant expresses a number of different enzymes that allow CBGA to differentiate into THCA, CBDA, CBCA. And then as we go through the process of decarboxylation by way of heat, then you get the more active forms of delta-9 THC, also known as the THC that we know and love, CBD and CBC respectively. We also know that there are also other cannabinoids. We have, instead of THC delta-9, we have delta-8. It's just a different isomerization of the delta-9. We also have delta-11. Delta-11 is not naturally expressed in the plant, and where you get that is when you mainly ingest any kind of edible form, and then your liver is going to convert that delta-9 into delta-11. Why is that important? Well, we'll kind of get, again, this further gets to the point of bro science. A lot of people go out and they make their medibles, and they don't test them. They just say, yeah, this has probably got 10 milligrams per dose, and it's probably going to be good for sleep. Yeah, let's just go ahead and mass market this accordingly. Well, what happens is when you ingest those medibles, that THC delta-9 is going to be converted into a much more psychoactive THC delta-11. And it's kind of a double whammy. So you take in through ingestion, and it has a much more delayed response. So here you are, you're sitting on the couch, you're watching, let's say, House of Cards, and you're just crushing these edibles. And you're waiting, wow, I'm not feeling anything, so I'm just going to eat more. And so that delayed onset is going to hit you, and it's going to hit you very hard because now you have a much more psychoactive form. So that's where you have to crawl out of the primordial ooze of bro science, get your products tested so that you know what you have. I always give the analogy that we're talking about medicine here. No one would doubt that. So why are we treating it like it's not medicine? Would you ever go into Walgreens? Would you ever go into Walgreens and they had a bucket of bulk pills, purple, red? Yeah, I have hypertension, just going to grab a couple handfuls of this and let's hope for the best. Yeah, nobody does that. Pharmaceuticals are supposed to be a targeted therapy. And what happens, obviously, you take pill A and then you have side effects, so you need pill B for the side effects of pill A, and that's basically how it works. But it's a targeted therapy, and that's where we need to go with cannabis. So getting back to the cannabinoid biogenesis, apart from the isomers there of THC, CBN. CBN, there are no high CBN strains of cannabis. CBN is basically the result of oxidation of either THC or CBD. And so one thing that we found is a lot of people are gravitating towards a product, maybe it's a tincture that's high in CBN, and it's a really great sleep aid. What you need for that is you either need time for that natural oxidation to occur, or you can expedite that process using a reducing agent, something like liquid nitrogen. We're seeing a lot of people having a lot of success with that, if you want to convert over into CBN. And then one of the lesser known compounds here is CBL, and that can be produced from CBC as a function of UV exposure. So, we get into a lot of this targeted use of cannabis, and if we know how to get to our target molecules, then we can go about things, intelligently. Okay, before I get into some of our latest research here, I just want to talk about optimization. Important issues. It's all very much data-driven. For me, I also have a tremendous background in environmental chemistry, done some work in that field. Looking at your environment is unbelievably critical. I think we've heard from a number of experts so far where they talk about nutrients and soil and whatnot. Water is really a big issue, I think, that is overlooked. Water is so unbelievably critical to all life forms that to just think, well, we'll just use whatever water and hope that it works out, I think is rather naive. In Arizona, we have very, very hard water. We also have water that's rich in iron oxides, and iron oxides tend to chelate arsenic. Now, as I said to you before, where arsenic was being used in a treatment for chronic myeloid leukemia, you're talking about parts per trillion of arsenic trioxide in conjunction with imatinib. But when we talk about groundwater that has 50 parts per million in some cases arsenic, this is just horrible water that has to be treated over and over and over again. After that treatment, it's not optimal necessarily for your operation, and people just don't know that. Treatment of water can come with chlorinated solvents many times, and you don't want that in your water either. So, you have to look at all of these variables. If you want to be growing premium cannabis, world-class cannabis, you have to know every single step. It's absolutely critical. Now, other things one thing I'd like to touch on here, and these are just thoughts that I'm throwing out there, things to kind of stimulate thought, but as this research continues to evolve, we also need to look at things like epigenetics. Epigenetics is a kind of a new field of genetics in that you can really understand how genes are expressed and how they're regulated. And this was best illustrated in a murine experiment where they took a series of mice and they made one of the mice diabetic. Okay, so the mouse wasn't diabetic previously, you fed it a bunch of pancakes and corn and all that good stuff, and now it's diabetic. Well, its offspring then was diabetic. Its dad wasn't originally diabetic. Through environmental conditions, it became diabetic, and it passed those genes, or the expression of those genes, onto the subsequent generations. So where I'm going with that is, if you don't know how you're treating your plants, and if you aren't treating them with love and care and if they're not your babies, then chances are they're not going to perform well, and in the subsequent generations, they're going to carry on, those epigenetic changes, and they're not going to perform well ever. So it's important to know every step of the way. Again, if you're in the quest of excellence and to provide premium cannabis to all of us who love that, you need to have every step absolutely dialed in. Now, there are a couple other things that we can look at here in terms of monitoring some of the changes. We heard from the guys up in Oregon there. We heard from a good friend here, Adam Jocks. We heard from Christian West. And those guys are more or less the Galileos of cannabis. And what they're doing is they're using science to track the progress of their plants, and then they're making intelligent decisions based on data. It's an incredibly novel thought in a field plagued by grow science, but it makes common sense to the rest of us that are trying to attack this from a more traditional angle. Another thing that we're trying to understand is endogenous versus exogenous cannabinoids, and I just picked THC for instance. So endogenous meaning that you have these cannabinoids that are being produced in the plant matter, and then exogenous is on the outside, or would be in these trichomes. So if we can understand the difference between cannabinoid production in the plant and also in the trichomes, then perhaps we can figure out a way to modulate that expression. For instance, we talk about people doing juicing. What if there was a way that we could determine which genes expressed or were turned on to enable endogenous expression of cannabinoids? We could push that equilibrium, and now all of a sudden you have a highly potent juice just from plants. These are the type of research questions that we're just starting to ask, and I think that there's going to be a lot of exciting discoveries in the near future. So I'm going to get into the data now. So this actually started last year. My friend and colleague here, Ryan, and I started C4 Laboratories, and I made a point that it was absolutely critical that we become heavily involved in cannabis research. So we started what's called the C4 Cannabinomics Collaborative, and it's a collaboration with my good friend and colleague, Dr. Kevin Shug at the University of Texas at Arlington, where we're using a $13 million analytical facility called the Shimatsu Center for Advanced Analytical Chemistry. It's absolutely world-class, one of a kind. And with those instruments in there, we're actually able to dive into this cannabis and really get our hands dirty. So the general thought was, okay, we're going to take cannabis, we're going to screen it using a number of different techniques, we're going to discover new molecules. Maybe we go in and we discover the structure of those molecules, for instance, with NMR or X-ray crystallography. But more importantly, we use a lot of based on gene expression profiles, then we can figure out what those molecules, what response they are eliciting, and then we know exactly whether or not that compound can be used as a therapy. And if we want to take it a step further, once we determine what those compounds are doing and the mechanism through which they're eliciting their response, we can figure out how to maybe optimizing it. So if we know which, for example, which proteins interact with these molecules, we can look at binding assays and we can model, and essentially we can take our original understanding of that structure and what it does and perhaps optimize it with a derivation. So once we figure out what's in there, what it does, the structure of it, how it works, then we can work backwards and figure out which of these strains has those molecules, and then we'll try to cultivate those strains in subsequent generations so that they can express more of that compound, thus being a more potent therapy. So, I'll talk a little bit about some of the putative cannabinoids here. Obviously, we have THC, anti-inflammatory, analgesic, neuroprotectant. It reduces intraocular pressure. This is great for people with glaucoma. Spasticity and muscle tension. We have CBD, fantastic molecule modulating THC-induced psychoactivity, anti-tumorigenic activity, and it's a very potent anticonvulsant, THC. And it's an incredible appetite stimulant. You see people that are afflicted with HIV or other viruses, it absolutely ravage their body. they need something like this that can trigger an appetite so that they can sustain themselves throughout their treatment. Also, incredible anti-tumorigenic activity. CBC, antibacterial, antifungal. Wouldn't it be wonderful if we could go into a hospital setting and people didn't have to deal with skin infections by Pseudomonas aeruginosa or MRSA? We could do that perhaps with a topical CBC treatment. We have CBG, excellence for inflammatory bowel disorder. Potent antibacterial, anti-tumorigenic. There seems to be a consistent profile here as a tumor therapy, as an anti-cancer therapy. Other ones that I'm particularly interested in are these two right here, these derivations. So THC-V is a propylated form of THC. And just the same, you have the propylated form of CBD, which is CBDV. Now these are incredible energy stimulants and metabolic activators. Also improves glucose tolerance and a potent anti-inflammatory. So as someone who's done a lot of work in Texas and lived there a long time, where macaroni and cheese is considered a vegetable, I think we would probably be in pretty good shape if we could use something rather holistic, plant-based, to help those folks. Who eat lots of their vegetables. Okay, so get into some of the structures here, and these are the molecules that we are screening for, and I'm going to get into some of the molecules that we're now discovering. So you can buy standards for all of these. So you have your THC, CBD, CBC. My understanding is we have THCV. We can do all of these in-house. Now the B squad players. I say that, kind of jokingly. A lot of people thought that Steph Curry from Davidson University was a B squad player, and we know how that turned out. I think the same is the case here. So CBDV, we're also looking at CBL, CBE, CBT, and iso-THC. You cannot buy standards for these, but we found a way to synthesize them in-house and use those as a standard. All right, terpenes, my favorite. This is where the aroma, this is where the flavor comes from. And made some good contacts with some new friends here who are making a cannabis-based beer. It's a great idea. Why does cannabis-based beer work? Well, cannabis is the closest genetic relative to hops. And now everyone wants to drink beer that's 99% hops and IPA central. And so it seems like there's a natural fit there. So in our lab in Arizona, we screened for, I think, 21 terpenes. And these all have a very similar characteristic. There are a lot of anti-inflammatories, a lot of antimicrobial. Interesting little fact. So that's where the genesis of IPA actually came from, is that they needed a way to preserve the water, so they treated it with hops because it had all this antibacterial properties. So those are the basic, and you've got things in there like eucalyptol. It kind of smells like a eucalyptus tree. D-limonene, you can imagine, citrusy smell. Alpha and beta pinene, you have that pine kind of tree smell. Now, here are some of the structures, but we're also getting into some of these new molecules that we're starting to see pop up in here. And you'll have to excuse me, some of these are even to pronounce for someone with a PhD in chemistry. But I'm going to try, so bear with me, I'm Canadian. So you have the lanolol, very nice lavender, reminds me of my wife making a fresh load of laundry that I forgot to put away. But what about exofenchol? What does exofenchol do? What is it? Until about 2 months ago, we didn't know that it was quite a prevalent terpene molecule in cannabis. Now, we're getting into some really exotic molecules here. Start with agarosphyrol. And then we also have alloaromatendrin. And of course, if you have alloaromatendrin, you have to have alloaromatendrin oxide. We're getting into some really, really exotic molecules, which gets very exciting because we didn't know that they were there in cannabis, but we have a better chance of figuring out what they do because these aren't Schedule A. People have been working on terpene molecules from different organisms for quite some time, so we have a better understanding of what these things actually do, and we're closer to using these as therapies. We have things like trans-2-pinenol, sativine, and valencine. So very, very cool molecules that we're just discovering here.
Excuse me, Judge.
Yeah.
So, crazy question, but has anybody done any correlation and crossovers with skunk spray? Because to me, sometimes what I have smells like skunk. Are there terpene crossovers with that aroma?
Ooh, that's a good question. I don't know. Where do they? We're just getting to the bottom of this, or we're just getting started here. And one thing I would add to that point is all of these discoveries have been from one graduate student in the last 3 months, moonlighting. We actually do not have funding to support her for her full PhD. She's actually doing this on the weekends. So, one, that illustrates that we surrounded ourselves with very, very smart people, and secondarily, that leads me to believe that the treasure box is full of treasure, and we just have to spend the time to dig in and find it. It's there though, and that's what's really, really exciting for us. So to answer some of your questions, we definitely would like to get down the road, but I mean, there's just a bajillion questions that need to be answered, but we're working on it. How are we doing this? So we run standard methods, ultra high performance liquid chromatography for cannabinoids. We use headspace gas chromatography with a flame ionization detector for terpenes, and a lot of the solvents, ICP mass spec for metals, and then we also use a PID, FID for fungicides, growth regulators and whatnot. But those are for our in-house lab. For the facility in Arlington, we have a lot of weapons in our arsenal. One of them is the VUV spectroscopy. And VUV spectroscopy operates under vacuum ultraviolet light. And with spectroscopy, you're actually able to tease out molecules that you can't differentiate in mass spectrometry. Why is this important? Well, because as you saw, we have a lot of different isomers that could have the same weight, same charge to mass ratio, but we can't really tease them out which is which, and they might have totally different functions and elicit different responses. So we had started teasing things out. Some of our environmental research, we were actually able to differentiate between NP and O-xylene isomers, which is cool. And now we translate that into the cannabinomics work. Another very cool thing is what's called SFE, SFC, MS. This is supercritical fluid extraction, supercritical fluid chromatography, supercritical. This is one of the only instruments in the world built by Shimatsu. It's a gorgeous instrument. And what this allows you to do is analyze some very, very volatile molecules, because you actually can take your nug, put it into a chamber, you put your chamber onto the instrument, and does the extraction in situ. And why this is very, very important is because when we're dealing with these cannabinoids, they are volatile, but they are not super volatile. Whereas the terpene molecules, if you open up your bag of cannabis, or let's say your bag of hops before you're about to brew beer, those molecules are volatilizing as you're looking at it. That's what they do. That's why you get that amazing aroma, because it gets into the air and they're very powerful molecules. So when you're doing an extraction in a tube, you can actually lose some of those incredibly volatile molecules before you actually get it into the instrument. That's just the inherent nature of this chemistry. And with this technique, we're actually able to avoid that. And I think we're going to find some unique compounds in there as a result. Last little tool in our toolbox there is negative chemical ionization mass spec. So with this is really good at looking at halogenated compounds. And generally you've got halogens you would associate maybe with pesticides, fungicides, growth regulators. But we've actually come across some natural cannabis products that have been brominated. So halogens include, fluorine, chlorine, bromine, iodine, that whole group. So for us to find naturally occurring brominated compounds, which we're further characterizing now, is very exciting because to the best of our knowledge, there wasn't any halogenated compounds, especially brominated compounds, to be found in the literature. So again, if I may come full circle here, what we're doing with this research, and again, one of the beautiful things about cannabis and cannabis science is that it's very inclusive. We have people that just want to know, they want to push the quality of the product, they want to help more people, and that makes it obviously very rewarding for us, but it does open some doors. It's quite contrary to cancer research where you got one group working on it on a new paper, and if another group who's working on the exact same thing publishes before this group, then they don't get paper accepted, and it gets very, very contentious. So it's not like that in cannabis, and basically our thought here at C4 and with our Cannabinomics collective or collaborative, we're open source. So if someone wants to come to us and wants to work, how it's worked in the past is you've had a lot of people come and they've brought us product, which we've been able to analyze, and any new material that we're able to analyze, we're able to learn more and get closer to the answers to these important questions. So, again, we're taking this cannabis flower, and we started on about 50 strains, and we're learning a tremendous amount, but there's obviously hundreds and thousands of strains that we could be analyzing and dedicating our lives to this. So we're finding new molecules, we're finding this treasure, unlocking these secrets. The next step is going to be to take some of those molecules, once we've established what they are, and then to figure out what they do. This is something that I'm very passionate about, and I want to take what I learned in cancer research to this level. One nice little anecdote that I always give, I can go all day about talking about the therapeutic implications, the social implications of how this plant is touching the lives of many. If I may talk about the financial implications though, there's an excellent example, a researcher at Harvard, he found one compound called resveratrol. Resveratrol was a huge boom to bust story in which it was extracted out of red wine. They found that it worked fairly well at extending the life of single-cell organisms. They then took that knowledge and they screened it for how this compound would do in mice. And the mice did really, really well when supplemented with resveratrol. At that point, I believe it was Merck or Pfizer bought that company out for $600 million, and later found out that resveratrol really doesn't do much what they thought. So unfortunately, that one didn't pan out. That's one compound. We're just scratching the surface with this, and I think there's hundreds, maybe thousands of compounds that we could really figure out what they do. Not only cannabinoids and terpenes, flavonoids, antioxidants. There's just so much in there. So after we figured out what these compounds do on a molecular level, then we can look at and we can assess whether or not we can optimize that therapy. Now, people talk about, well, okay, is it best to extract out those compounds of interest and make them 100% pure? Or let's just make a synthetic version and it's 100% pure, just like the pharmaceutical approach. We don't know. The truth of the matter is, sometimes that might work, sometimes it may not. What we do know is that there's a lot of interaction between all of the cannabinoids, all of the terpenes, and they work together in synergy. That's why the plant expresses them, because they work together for the plant, and coincidentally they work together for us. So that's again something we're trying to get a handle on. And then once we find out how these work, then we can work backwards in optimizing strains and pushing the equilibrium in that genetics for particular molecules. It's been my opinion, there's been a lot of great work done moving away from THC now into CBD, but I think there's going to be another shift maybe down the road looking at CBC, and I would love to see someone tackle THCV and CBDV. I think there's tremendous potential there, but it's going to happen. I mean, it's just everyone has their hands full, and we're trying to burn the candle at both ends. There's just too much to take on. So that is my spiel, and I appreciate your time and consideration. I'd like to open it up for any questions.
I've heard that THCV is an appetite suppressant. Is that true?
That may in fact be true. The literature shows that it's an incredible metabolic stimulant and actually can antagonize beta oxidation, which is a way to break down fats. So I'm looking at it as perhaps we make a THCV-rich supplement that is like the cannabis version of hydroxycut. Maybe we do that, or maybe we grow THCV-rich strains, and we don't have to feel so guilty after we just crush 10 pounds of turkey after Thanksgiving. We just have our little THCV, and we're doing a lot better.
And are you able to do this research because you've broken it into molecules and you're not researching cannabis? I mean, vis-a-vis the federal law.
Sure. So through our collaboration with UT Arlington, we are operating under a DEA license, and so that's definitely opened some doors. It's also important to when we send a lot of extracts as well. And when you do an extract from flower, we need to make sure that the THC levels are below 1 milligram per milliliter. And we also have to look at, okay, well, you're extracting into ethanol. Ethanol is flammable, so what do you extract into? Well, then we have to consider that as well. So there are a number of roadblocks, even in trying to transfer these samples to do this research, but we're getting it done, and we're making some serious progress.
Thanks, Zach. Good presentation. I have a question more of a resource. So,
Sure.
One is a protocol for dosing, for, like you said, for HIV/AIDS patients. Specifically, my question is interactions with drugs in pain management for chronic pain patients.
Yeah.
So, some very well-respected neurosurgeons and pain doctors are concerned with the interactions, for example, with ketamine, so cannabis and ketamine. So the interactions and the dosing for those patients, just like you said with edibles, do you have a resource, or is that something that you're working towards? I know there's projects you do that seem to be.
Yeah, we're certainly working towards that. There's so many questions that need to be answered that are patient specific, and I think the research is just getting started, and it hasn't. Unfortunately, we haven't had the resources at our fingertips to really push that equilibrium, but I think we're very close. As for that question about HIV, I found a lot of people are doing very well with exogenous hormone therapy coupled with cannabis. So they're using a lot of testosterone therapy. They would probably drop the ketamines and any kind of heavy narcotic and go into cannabis, and they're doing very, very well.
Which strain?
Oh, I don't know. I mean, this is, again, just, you're getting into the preliminary literature. People are writing editorials. None of this is peer-reviewed yet, and so I would characterize it as anecdotal. But, these are an N of 1 or 2 that, some physician may be observing.
Are the terpenes in cannabis specific to the cannabis plant and not available in other plants, or is there?
Oh, that's a great question. So there are some that are specific to cannabis, some of those exotic ones that I just put up there. But most of them, are found in other compounds. I mean, hops, for example, closest genetic relative, tons of terpenes, and there's a lot of overlap there, but there are a couple, and as we discover more, I'm sure we'll find some that are unique to cannabis.
It appears that recent research is indicating that the terpenes are having a definitive specific effect on different individuals, and the percentage of those terpenes in the respective strains is something now that I guess people are taking it out in the extract and then adding it back in different percentages to have different effects.
Yeah, I mean, you're definitely trying to optimize it, what I would call in vitro at that point. I've always preferred in vivo, And what I mean by that is not pulling them out and putting them back in, but I think they're pretty therapeutic on their own. Some of the best cannabis that I've ever enjoyed has been not relatively high in cannabinoids, but just loaded with terpenes. And it's just the way that it was grown, the way that it was preserved. Maybe have a 5 or 6-fold terpene concentration than your normal average Joe cannabis, and it's amazing. And it's because of that synergistic effect of all those terpenes coming together. So that's what's really exciting, because I think there's a lot more terpene molecules in there than people realize, and we should give them a little bit more respect. You know, we're giving THC respect, we're giving CBD respect. Well, those terpenes are important as well.
What forms of extraction methods and solvents?
Okay, so major solvent is just methanol. It's pretty good from my experience. We've messed around with some other solvents, but you know, we hear people using other alcohols, but no, it's pretty simple. Methanol works.
What about THC, like as an acetate? Do you have any studies about that at all?
No, great question. We have not dived into that. Yeah.
Do you foresee in your studies the determination, the conclusion that the growing environment will have a great effect on the output of the plant and its benefits?
100%. And that's going to be through those epigenetic pathways. So that plant, it's certainly going to come back. So let's say we're using bad water and we're depriving it of water, and so it's wilting a little bit, but then we bring it back. I believe that you won't be able to see that change in the genetic code, but it is happening on the molecular level. So we should just treat our plants with respect every single time. That will carry over into subsequent generations.
Is it mostly is it mostly marketing when people say something to the effect that certain cultivars are high in CBD or THC or certain constituents?
Yeah, there's actually two problems there. I mean, and again, coming from more traditional medicine, I mean, people come out with these strain names and the nomenclature, I can't follow it, and then it's like, again, bro science, like, oh bro, this is what I've got.
Right.
And dog OG, and then he takes it home, like, yeah, I got this strawberry OG Kush from
Right.
this guy. And it's like, how do we actually know?
That's a big question that I've always kind of asked.
I worry about that all the time. So we have heard it come in and people say, oh no, my blackberry Kush always measures at 17, every time. And it's like, I don't think so. It's going to vary from plant to plant in the same grow, let alone from subsequent grows.
I've heard that there's a group that is creating THC from yeast.
Okay.
Oh, I've heard of that too.
Sure, absolutely. So they figured out the enzymes that can express these compounds and do the biogenesis in vitro, and you've got yeast now producing these molecules. And I think that definitely has some promise, but now we're back to, okay, we grow up all this yeast, we lyse the yeast cells, now we extract this THC, and we just have what I would call a biosynthetic, I mean it's recombinant, but it's a biosynthetic version, right? Just like a synthetic version you make through organic chemistry. Whereas what we're finding is that, again, give those terpenes some love. They're important. Let's go through the plant and extract it, with all of its little buddies, working together. Sorry, I'll get back to you.
At the AOCSA meeting last week, someone presented on proposing a different type of classification system using terpenes and chemical footprints to classify different types of strains. Is that possible?
Well, sure. I think because you have more terpenes, and so it's like a much broader signature, if you will. That's going to take a lot of work. That's DNA, whether it's you look at kind of classifying things based on their phenotype, how they express those genes. It's going to take a long time, but I would advise them to get started. And maybe that would alleviate some of the concerns with, I have ChemDog X or whatnot and totally mischaracterizing it. If we actually know how this plant is performing based on its signature, then we know exactly what it is and we can just work backwards. Yeah, exactly,
Right,
exactly. Because
right.
obviously you can't do it through phenotype. You can't just look at the plant and know.
There's a lot of debate about which environment grows the most full spectrum plant profile. Indoor, outdoor, soil, organic, synthetic. Do you have an opinion?
As someone who can't even grow one plant, never done it, never had any success. I have no idea. We have a guy over here who knows. But yeah, no, I can't really comment on that. But that's where I think it's very exciting. You can take a guy like me, collaborate with a grower, look at how the environment is affecting the product, and then proceed accordingly. We obviously heard about how indicas versus sativas need to be treated differently. Nutrients, metals, soil, water. So we should really follow that if we really want to get it out in. Yeah. Yes, sir.
Exactly. We talked earlier, I use clear oil for my extract.
Yeah.
So winterize, distill, terp. At that point, does the strain or even indica versus sativa matter once it's gone through that much processing?
Yeah, I mean, if you're deterping, essentially you've lost all those flavoring components, but you also have a variance in the cannabinoid profile. So you might have a different ratio in CBD to THC, you might have different CBG, different CBN, THCV in there. There's going to be some minor variation. Yeah. Somebody else has a question? Yes?
Let's say a client works with your laboratory.
Yeah.
What's the cycle time or lead time? What's the turnaround time actually?
You want to analyze some product? 24 hours.
Wow.
So I ship FedEx.
Well, we can't do that. Where are you coming from?
It's a trap.
California. No, what we're investigating and trying to do with out-of-state clients is to give them a kit where they can do the extraction themselves and then send it to us, while keeping the THC concentration below that certain level, and then also making sure it's not flammable. It kind of handicaps us, but that's just the nature of the beast. But yeah, we turn things around in 24 hours because, as we find, a lot of the producers and these dispensaries, they have something that's really, really good. They can't wait a week to get that data back. They want to know what's in there right away because they're going to sell it very quickly. So we need to respond to.
If I may add to that, the quantification of cannabinoids and terpenes, those things are a relatively quick runtime, and there is a volume limitation to that 24-hour turnaround. It's about a 30-minute process to identify those residual solvents and terpenes. So obviously once you get above the 40-45 samples in a day, then obviously the 24 hours is a very tall task to fulfill. But generally for those basic analysis, we can do 24 hours. The more complex analyses, such as the pesticides, fungicides, growth regulators, full mold counts, and those type of things, is more from the 72 hours to maybe 4 days or so.
Can we get up there, big guy?
No, I just wanted to clarify and not overpromise there.
Any other? Yes, sir.
Yeah, just going back to your statement about epigenetics and the viability of the plant and generationally, wouldn't you say though that an organic plant is probably going to be epigenetically more grounded in its genetic roots than something that maybe was raised chemically?
We have no idea. That sounds like sound logic, but we have no idea. People just started doing epigenetic studies on mice and Drosophila 4 or 5 years ago. And what we're talking about epigenetics is basically that DNA becomes methylated, and if it becomes methylated, then it's no longer going to be expressed as RNA and then turn into a protein. So I don't know. That's what I want to find out. To be honest with you, I'm in a position here where I'm working with this great team, and I know that there's treasure in that box. And we just need the resources to get in that box and to find it. It's there. And that's what I'm excited to embark on. Yes, sir.
You said there's multiple molecules
Well, okay, yeah, that's a great question. So, again, what we do is the thought process is here, we discover what's in there, okay? We find a unique molecule, we can extract it, and if we really want to study what it does, what we can do is we can figure out what this thing does on its own. You know, how does it affect gene expression on its own if we chemically synthesize it? Then, what does it do on its own, or what does it do when it's in mixed with all its fellow cannabinoid buddies and terpene molecules? And then we can further understand, you know, what is really going on. Because as you alluded to, when you just pull out one compound, it's really kind of an artificial environment. That's not how it works in the cell. But what we can do is we think that, okay, this does a lot of good, let's say as a metabolic stimulant, with all these terpenes, let's somehow push the equilibrium to ramp up that expression. So instead of 0.5% THCV, you've got 4%, something like that. Yeah. Any other questions? All right, well, thank you very much for your time. So let's have this day.